Hello students, so we will continue with the previous chapter, that is heredity and evolution, and today again we will discuss about some of the basic terms in biology, okay, that is in molecular biology or genetics, that is what are chromosomes, chromatin, gene, DNA, replication of DNA, replication of chromosomes. So we will discuss about all these basic terms in molecular biology, and then after that we will start with the heredity and variations. As I told you in the previous class that the chromatin, that is a thread-like structure, highly coiled structure, which is found in the nucleus of the cell. Now this chromatin, just before the cell divides, it condenses, means it highly coils and it shortens and it forms the chromosomes. Now chromosomes you can see here in the structure also, and we have discussed this again in the previous class also. Again once I would like to repeat it. This chromosome is unduplicated chromosome, as you can see in the slide. There are two chromosomes shown. One is duplicated chromosome and another one is unduplicated chromosome. Duplicated chromosome I will tell you later. So this chromatin, it condenses to form this unduplicated chromosome. Now actually what is chromosome? Chromosome is nothing but it is DNA, that is deoxyribonucleic acid. Again you can see this figure or this picture, that is chromosome DNA molecule. Here you can see that cell is there, and in the cell nucleus is present, and in the nucleus chromosomes are present. Now these chromosomes I am talking about unduplicated chromosome in the undividing cell. Now this unduplicated chromosome is nothing but it is DNA. You can see here also in the picture also you can see that this is DNA. So chromosome is nothing but it is DNA. Again you can see the structure of chromosome. You can see in structure of DNA. What is DNA? You can say the chromosome as DNA also, or this one single DNA you can see here, this you can call as chromosome also, okay, but the chromosome here is unduplicated. Now in structure of DNA you will see that there are two side strands, and these two strands they coil around each other, that is DNA, and these two strands are called as the backbone of DNA, and these strands are made up of these strands are made up of sugar and phosphate. No need to remember the chemicals of which they are formed, but just for your understanding I am telling you these strands are what actually they are made up of sugar and phosphate. And in the center you will see that there are horizontal rods. These horizontal rods are the nitrogenous bases. You just see in the DNA where the two strands are coiled. So it is DNA, deoxyribonucleic acid. It is made up of sugar, and the sugar is deoxy sugar. It is made up of phosphate, and it is made up of nitrogenous bases. Now when you will uncoil the two strands, it will give you the appearance of a ladder. The side portion of the ladder, that are the two strands, and these horizontal rods in the ladder, the steps which you can see are actually the nitrogenous bases. So this is DNA. Now what is gene? Gene are not some stars like things which are attached to the DNA. Genes are nothing. Like you all know DNA now, just now I explained you. In the DNA there are certain portion which is functional and certain portion which is not functional, and these functional portion is called as the gene. These functional portion may include the nitrogenous base and the backbone, that is the sugar and the phosphate. Okay, students, these two strands are also called as the backbone of DNA. So this gene is the functional portion of DNA which will include the nitrogenous bases as well as the portion of backbone, that is the sugar and the phosphate. So this gene, that is the functional portion of DNA, it synthesizes a particular protein, and that particular protein controls a particular character. So in the DNA there are many functional and non-functional portions, and all the functional portion is called as the gene, and there are different type of genes, okay, and these different types of genes, they help in the synthesis of different proteins which control different characters. There is no similar gene in the DNA. Why, what, that, all these things you will study in higher classes, not now. Okay, again you can see the diagram of gene here. You can see this is cell. In the cell, chromosome is present. What is this chromosome? This chromosome is actually the DNA, and you can see here gene is marked. This is the functional portion of the DNA, which will include your strands, that is the backbone also, as well as the nitrogen base also. Okay, now gene is the functional portion of DNA, is called gene. It provides information for the synthesis of protein that controls specific characters. Okay, thus gene is considered as the unit of heredity. Why it is the unit of heredity? Because it controls the particular character by synthesizing proteins. So this gene, this is the gene which goes along with the chromosome to the offspring. And here, if the same gene goes, the same protein is synthesized and it controls the same character in the offspring as it in the parent. So this is also called as the unit of heredity. Thousands of genes are present on the chromosome which control various characters in organism, and there are around 30,000 to 40,000 genes in 46 human chromosomes. So all your characters, everything is controlled by the gene, or all your information is present in the DNA. Okay, now I will come to DNA replication. Now students, this is DNA, as you can see. This is DNA, you can say, or this is unduplicated or one chromosome. Now what happens just before the cell starts dividing? The DNA, two strands of the DNA, it starts uncoiling. Uncoiling means it starts separating. And when it separates, okay, now it is not when whole of the DNA will separate. What happened? It starts separating, and that portion which is uncoiled, at that portion, in both the strands, in both the uncoiled strands, a new single strand of DNA starts forming. Okay, as you can see in the picture also, whole of the DNA, strands of the DNA will not uncoil. It will start uncoiling, and rest, and the portion or the strand which is uncoiled, from there the DNA synthesis will begin, or you can say from there one new strand of DNA will form. And as this formation of new strand progresses, the rest of the portion of the DNA, that is the parent DNA, also uncoils. So here what you are seeing, that in the old strands, the new strand starts forming, and this new strands again coils with the parent strand. So now you will see that there are two exact copies of DNA is formed. Okay, in the two strands of DNA, two exact copies of DNA are formed, and in one copy, or in each copy of these two DNA, one is the parent strand and one is the new strand. I hope it is clear. Okay, you can even see it's very clearly shown in the picture that the two strands have uncoiled. Okay, and one new strand is forming, which is coiling around the parent strand, and it gives the appearance, and it is the DNA, and these two DNA are exactly similar. Okay, this is called DNA replication. DNA replication happens just before the cell division, and here the two exact identical copies of DNA is formed. You can see a replicated chromosome, which also is called as a duplicated chromosome, or duplicated DNA also I can say, contains two identical chromatids, also called as sister chromatids. A duplicated chromosome therefore includes two identical strands joined along their length to corresponding length of DNA. First of all, you can see here that now what is formed here, two identical DNAs are formed. I can also say these two identical DNAs are like two identical chromosomes. Okay, so now what happens, these two identical DNA, okay, you can see in another picture, here you can see replicated chromosome. Here you can see first it was in A, this is unduplicated chromosome. Okay, now this unduplicated chromosome has only DNA. Okay, now after replication, what has happened? Just now you have seen, now there are two exact copies of DNA, and these two exact copies of DNA are joined by a structure called as centromere, as you can see in B, figure B or picture B under replicated chromosome. Okay, so after replications, two DNA strands are formed and they are held together by a structure called as centromere. And in each of these DNA, we can call it as a chromatid. There are two DNA exact copies. So one DNA is called as one chromatid and another one is called as another chromatid. So in a duplicated chromosome, there are two identical copies of DNA and which forms chromatid. So there are two chromatid. One chromatid, one copy of DNA, and that one copy of DNA will contain the parent strand as the newly synthesized strand, and another is the another chromatid. It contains another copy of DNA. Okay, so in the duplicated chromosome you will see two chromatids and they are called as sister chromatids because they are exactly similar to each other. So students, today we will do till here, and in the next class we will start with mitosis and meiosis. I will not tell you in detail what is mitosis and meiosis because it is not required in class 10th, but I will, in a nutshell, in a summary, I will tell you what is mitosis, what is meiosis, and then you will start with heredity and variation. But just go through whatever I have explained to you, no need to go much in detail, but you should know what is a chromatid, what is centromere, and what is a duplicated chromosome, DNA, all genes, all these things you should know. Okay, thank you students.